So this is like collection of points which we had seen before. And that is it. These issues and debates are only in Freeman. So this completes the Freeman study. Do you have any doubts? Okay, then I'm coming back to the textbook. Okay, in the textbook, next thing we have to see is explanations of schizophrenia. Any disorder that we have, first they explain, they give us the symptoms and all. Then they consider why does the disorder take place, what are the causes. So that we see under explanation. Now they've given us three different explanations. The first is biological. So we have to take one by one in turn and keep studying and evaluating. Pattern remains the same. Lot of boxes are there. Directly we come to page 242 and we come to genetic explanation. This is the first one. Okay, now when it comes to biological explanations, there are genetic and biochemical explanations in all disorders. Now here we have to do good amount of memorization. So some points I'll mark in pink. As they are given in the textbook, we have to memorize for the paper. Now here they have told us something about monozygotic and dizygotic twins. I'm just marking the point first and I'll explain something. Okay, there are two types of twins in this world. One are monozygotic twins. They are also called MZ. Other are dizygotic twins. Now in common language, monozygotic twins, we call them identical because they look the same. And dizygotic twins, they also either they look same or similar, but we call them non-identical, or we call them fraternal sometimes. They don't look exactly the same. Some differences are there. Now we have to know what is the difference between them from a genetic point of view. Okay, when it comes to monozygotic twins, when the twins have been conceived, that time the mother is, we know from biology, we have learned there is a zygote that forms in the mother's womb. So when she conceives, there is one sperm from the father and there is her own ovum or egg which combine and form one cell in her womb. This is same as if she is going to have one child. In that case also there would have been one zygote. But in the case of these twins, that one zygote after some time it splits into two. Now because of that, what happens is, because they are from the same zygote or the same first cell, their genes are exactly the same. It's just that one cell has split into two. So their genes are identical. So they are, we'll say 100% genetic sharing or exactly the same genes. That's why they're always of the same gender also. Either both are boys or both are girls. Because like we know, gender we get from genes. So that is why both have to only be of the same gender. Now coming to dizygotic twins, how they are different is when the mother conceives them, there are two different zygotes in her womb. So from the beginning only, there was never a single zygote, there were two, because her two different ova are fertilized by two different sperms of the father. Just coincidentally, it happened that at the same time. Now their genetic sharing, because from beginning only there are two different cells, is about 50%, not 100%. It's as good as if supposing I have a sibling who's a little elder to me, 3-4 years, or younger to me, whatever, 5, 6, 10 years. The way I share my genes with my sibling who's born at a different time, these two also share that much genes only. It's just that they're conceived at the same time. That's it. Now, why we have to know about this genetic factor is because considering disorders, supposing we are like right now we are studying schizophrenia. If I find that in most cases of monozygotic twins, when one twin in the pair has schizophrenia, the other also has, then I will feel it is because of genes, because their genes are exactly the same, so both must have got it in the genes. That is my logic. Now what happens is, when I give this logic that both were in the womb and they got the same genes, that's why they got. Now some people will argue, but maybe it is because the parents are treating them in the same way. They were born at the same time, so parents must be giving them same behavior, same schooling. In fact, we see similar clothing, everything. Maybe that's why both are developing the disorder, not because of genes. So then what we do is, we compare them with dizygotic twins. because even they are born at the same time. Now, supposing we see in the case of dizygotic twins, this is not true. When one in the pair has schizophrenia, sometimes the other one has, not always, not like monozygotic twins. Then we can say it is not because of the way parents are treating them, because parents are treating dizygotic twins also in the same way. Then even they should have very large amount of sharing of the disorder, which we are not seeing. So then it must be coming because of genes. So this is the basic logic which is underlying in all the disorders that we are going to see. Okay. So now let's come to the point which we have marked specifically for schizophrenia. First thing we have to memorize here is the name. Gottesman and Shields, though it is given in the bracket, the name and the year both we have to remember. These are the researchers who have found out what is the sharing rate for schizophrenia in twins. So in the paper when they are writing, they mention their name also. Concordance rate simply means sharing rate. So what Gottesman and Shields did, they took many pairs of monozygotic twins. All these pairs were such that one at least twin had schizophrenia. Okay, and then what they saw was in all these pairs, how many pairs are such that both the twins in the pair have schizophrenia? Means is it that in all the pairs only one has, or in some pairs both have? That they were studying. They found that 42% such pairs were there in which both had schizophrenia. So for simplicity, let's assume they study 100 MZ pairs. In all 100, we are seeing one is having schizophrenia. But out of these 100, 42 are such in which both are having. And only 58 are such in which only one is having. This is what they found. Okay. Now we know we can't stop here. We have to compare with dizygotic also. Otherwise, we'll feel it is because of the environment. So then they studied dizygotic twins also. Okay, all the figures are very important. So for monozygotic, we saw it's 42. For dizygotic, it is 9. So next what they did, they took 100 dizygotic pairs. In this also, in all 100, one was having schizophrenia. They saw in how many both are having schizophrenia. So there were only 9 such pairs. In 91 pairs, only one had. So clearly we can see in case of monozygotic twins, the sharing, or what we technically call concordance rate, is very much higher than in dizygotic twins. Like 9 versus 42. It becomes more than 3 times also in monozygotic twins. So this tells us that somewhere genes are playing a role. No doubt environment is also playing a role. Otherwise, how do we explain in the remaining 58? How come only one is having? But in the 42 in which we are seeing, there definitely it is showing us that genes is playing a role. Not 100%, it's not the only factor determining, but it's a major factor which is determining. So this was their conclusion. So the logic also they have explained, I'll just mark it quickly. Since the siblings in both groups were raised in the same household, like I said, they're given same treatment by parents, higher concordance rate in MZ twins shows that it's because of shared DNA or simply shared genes. Because as such, even dizygotic twins get the same treatment. Now these methods of studies, these are called twin studies. These are little outdated, so they come under classic research. This type of studies were done, like we can see in 1966 and all, when technology was not very advanced. So researchers didn't have other ways, like looking at genes directly or something. Now they have better ways of looking at genes. So now we have contemporary or modern research. Now they use scanning methods, medical methods, and those diagnostic type of physical diagnosis methods. With that, they're able to look at specific collections of genes which are playing roles in disorders. Like as long as we are looking at twins, we are saying, okay, genes play a role, but we can't say which genes are playing a role. Now they are finding out which genes are playing a role. Okay, what they found is that whole genomes of people tend to play a role in disorders. Whole genomes means groups of genes. Like when we are born, that time we have thousands of genes. Some are coming from the mother, some are coming from the father. Now, it is not possible to pinpoint and say, supposing gene A is playing a role in schizophrenia. It's not as simple as that, that only one gene is determining one disorder. It is a collection or combination of genes. So we call it genome. Entire sets of genes are playing a role. Now, these type of studies in which we find out which are the collections of genes which are playing a role, they are called genome-wide association studies. GWAS. We have to memorize like this only, the full name and the abbreviation which is given in the bracket. Like this only we have to write it in the paper. Now further they are explaining to us what they find in these studies. Some genes are polymorphic. Polymorphic, like they're telling us here, they come in different forms. Like say there is a gene which we will see about ahead, it is called COMT gene. Everybody has this COMT gene, all human beings are having, so we all are having it, but we all are not having the same version or the same form of the gene. So supposing hypothetically it's coming in three forms, A, B, C. Some people have COMT A, some people have COMT B, and some have COMT C, like that. So genes come in different forms or different modifications are there of the genes. That's why they are called polymorphic. Polymorphic means many forms. Sorry, are they? Yeah, they are alleles, correct. That is the scientific term. So allele just means version, form, modification, whatever we call it, but technically it is called allele. Now what researchers do is, supposing they've come to know, like we saw the example, COMT is in three forms or three alleles. So now they want to see, by chance, if a person has A allele, is he more likely to develop schizophrenia than someone who has a B allele? So they just want to compare and see. So what they'll do in the study, first they identify which gene the person has. They come to know from blood samples and all. So they find out, and then they will compare, like, okay, these, supposing 50 participants have A, 50 have B. How many of the first 50 have schizophrenia? How many of the next 50 have? Supposing in the A group, they are finding too many have schizophrenia compared to the B group. So that is showing A is creating a risk for the disorder. So it's as simple as that. Okay, so now they are telling us, like we were seeing earlier also, disorders are polygenic. Lots of genes are playing role. That is what they come to know in genome-wide association studies. So it's not only in schizophrenia, it's in all the disorders we are going to see, even depression and all. No one gene is playing a role, many genes are playing a role. So any condition like this, any mental disorder in which many genes play a role, technically it's called polygenic. Poly again means many and genic just means genes. Now they are telling us, when it comes to schizophrenia, they have found there is a neurotransmitter in the brain called dopamine. Genes which are responsible for dopamine, they are playing a role in schizophrenia. Like we know in biology, we study, we inherit genes, then whatever cells are made in our body, cells like we know are the basic units. These cells are made according to the genes which we have inherited. So the genes have the instructions for how the cells will be made. Now people who are having schizophrenia, their genes are not proper, like we're seeing something is problematic. So the cells are not getting created properly in their brain. So the cells which create that chemical, which is called dopamine, those are not getting created properly. So dopamine levels will not be like normal people. They'll be more or less compared to normal people. Okay. So they are saying what they've identified in the case of schizophrenia, three problem areas are there when it comes to dopamine. Those cells which are responsible for development or synthesis, simply meaning production of dopamine, they are not getting created properly because the genes are not proper. Or in the transportation, we'll see ahead, some cells, what they do is they play a role in transportation, taking the dopamine from one cell to another area where it is required. So those cells might not be working properly, means genes in production, genes in transportation, all are problematic. And finally, breakdown. Breakdown is what? Ahead also I'll show properly. When it comes to the chemicals which are getting released in the brain, they're always little more than what is required. So then the extra chemical which gets produced, it needs to get cleaned off so that the brain does not have any waste or unnecessary material. So So it needs to be broken down. So that extra waste chemical which is there, maybe it's not getting broken down properly. So those cells which should clear off the areas of the brain, they are not working properly. We look at all of this. Okay, so for now what we are understanding, cells which are producing dopamine, helping it reach from one area to the next where it's required, so transportation, or which are breaking down waste unnecessary dopamine. All are in problem in people having schizophrenia because their genes for dopamine are not proper. So this much we have understood. Now coming to specific genes. With time, what is happening is, as technology is becoming advanced, researchers are able to pinpoint genes also. Of course, collections of genes are playing a role. In those collections, they have identified some genes. Some, as research advances, they will tell us about more also, which are there in those collections. So for now, they are giving us the example of two genes, which they have found repeatedly that they are playing a role in schizophrenia. Okay, one is the COMT gene, which I mentioned earlier. Now, how have they come to know about COMT gene? We have to see that also. So here also there are some things we have to memorize. Okay, they are saying there is a condition which is called DiGeorge syndrome. So this we have to remember. Anytime anything in the textbook is given in bold, it is very important. So DiGeorge syndrome is there. Syndrome is not a full-fledged disease. It's like a condition in which person shows many symptoms. So it's very close to being a disease. For now, we can definitely take it as a disease only. So people who have this disease or this illness, we can say, which is called DiGeorge syndrome. In them, what we find, a full strand of DNA which has 30 to 40 genes is just deleted from chromosome 22. Okay, again in school we learn that we have chromosomes like this, with DNA which is intertwined. And we know that genes are the segments on the chromosomes. Now what they are telling us, in these people, in their chromosome 22, so supposing this is chromosome 22, one entire section of genes is deleted. Supposing this whole section is just not there of genes. So they are said to have DiGeorge syndrome. Now what they found is, in those who have DiGeorge syndrome, 1 in 4 of them, that means 25%, end up developing schizophrenia. Schizophrenia, we have seen, it starts in age of 20s. So right from birth they are having DiGeorge condition. By the time they reach 20s, 25% of them develop schizophrenia. So here we have to remember 1 in 4, or we can remember 25%. In normal people who don't have DiGeorge syndrome, only 1 in 100 people develop schizophrenia. So only 1%. So it is 25% versus 1%. Clearly we can see this chromosome 22 and the deleted genes are playing a role in schizophrenia. That's why their risk has increased so much. Now, out of these many genes that are deleted, one they have identified is COMT. So, in the people who are having this DiGeorge syndrome, the COMT gene is missing. This is very important to be memorized. Like when we are evaluating also, we have to give examples of genes like this. So, COMT, we can mark it as 1. This is the first gene they are discussing over here. Now, how is deletion of COMT creating a problem? They have explained that also. They are saying this gene, it codes, means it creates an enzyme which breaks down neurotransmitters such as dopamine. We were seeing first also breaking down is important. So, COMT, it creates some enzymes. Enzymes like we study are just proteins which help to clear off some chemicals in the brain. These enzymes, they help in the breakdown of dopamine. Now, because the person is having DiGeorge syndrome, his COMT gene is just not there in the 22nd chromosome. Because of that, that enzyme is not getting created in his brain, and because of that, the breakdown of dopamine is not taking place. Because of that, the extra dopamine which is there, which, like I was saying, it should get cleared off so that wastage, unnecessary chemicals are cleared off, it will still remain. Extra dopamine will remain in the brain. It will not get cleared off. And ahead we will see in the next explanation, it is found that people who have schizophrenia, they have more dopamine than necessary in some areas of their brain. That's why they get these hallucinations and delusions which we were seeing. So this is the explanation of why it happens. The COMT is not there, so dopamine is more, and so hallucinations and delusions are taking place. Okay, so this is one gene which they found. Like this, they found one more, DISC1. COMT is having some very technical name like catecholamine and all, so that they have not given. We don't have to remember. But DISC1 is having a simple common name, so we have to remember the name as well as the abbreviation. So it's just disrupted in schizophrenia gene, which is abbreviated as DISC1 gene. Okay, this also they found here. This is not related with D. George syndrome. This they have found in some other studies. They have not elaborated much. What they found is in people, this DIC gene is there, means it is present in them. It increases the risk of schizophrenia. In COMT, we saw if it's deleted, it's increasing the risk. DISC1 is creating a risk if it is present in the person. Now, how it is doing it? DISC1 is related to a neurotransmitter, like we can see, which is called GABA. This is another chemical in the brain, just like dopamine is one chemical, GABA is one chemical. GABA by itself does not create schizophrenia. So it's not like someone has more or less GABA, so he has schizophrenic symptoms. No. But GABA regulates or manages dopamine levels in the brain. So indirectly. What GABA does, we can see ahead and we'll have to remember for the paper. GABA is an inhibitory neurotransmitter. Inhibit, as we know, to stop something. So what it does is, when GABA levels are normal, dopamine levels are less. Means how much they should be. It manages dopamine in the sense it keeps dopamine at control levels. Supposing, let's say, if hypothetically in our brain we should have 50% dopamine at any given time, not more than that. Because GABA is there, the level is maintained at around 50%. Now imagine GABA is not there properly, because the DIC1 is not proper, means it is there and it is creating problems in GABA. Now GABA is not there properly. Because of that, instead of 50%, the dopamine level jumps to 75%. It becomes too much because GABA is not controlling how much dopamine is there. Again, the same problem. Dopamine is too much, so hallucinations and delusions type of symptoms will come. Okay. Yeah, it means GABA is becoming much less than what is needed. If it is too much, then dopamine levels will become lesser. So instead of 50%, say they come down at 25%. But that is not much of a problem in schizophrenia. So because now we are seeing schizophrenia, we are not looking at that problem. In schizophrenia, there's a problem if dopamine is more, not if it is less. Okay, so here they are telling us this is how DIC1 is playing a role through this mechanism. So they are telling us that is the abnormality of this gene. Again, here they have not given in detail, but DIC1 also exists in alleles like other genes. So which DIC1 the person is having, that also plays a role, but we don't get into all that. They are just saying abnormality of the gene. So they are talking generally. It increases vulnerability to excess dopaminergic activity. Means we can put it simply as more dopamine in the brain. Like we were just seeing. So because there's abnormality or there's presence of DIC1, that's why there's more dopamine in the brain, and that results in symptoms of schizophrenia like hallucinations and delusions. The positive symptoms basically come because of dopamine. Okay, this is all that is there in the explanation of the disorder. So for this also I'll give the notes in that. In brief I have given for a 6 marker how much we have to cover. Now coming to the evaluation. So before we see the evaluation, let's just quickly see in the textbook what they have given. One is all these biological explanations and treatments, always they have a lot of research support. A lot of research goes on in the biological area. So what they're telling us when it comes to the COMT gene, like I have given an example, there are variants like A, B, C. Now they have given us the proper names of those alleles or variants also. So far in research, they have found 2 alleles of COMT gene. So we can say alleles or versions. One is MET and the other is VAL. So instead of A, B, they have given names. No, COMT is the gene and it has versions under it. Okay. So COMT has one version of allele called MET and the second is called VAL. Now researchers use the same logic which we were seeing before. They see those who have MET, are they having more risk of schizophrenia, or those who have VAL? So they have done such studies. What they found is VAL is associated more with schizophrenia. So here we have to remember VAL. And again they found the mechanism. How is it increasing? So what they found is because of VAL there is decreased dopaminergic activity in prefrontal cortex, decreased cognitive performance and slightly elevated risk of schizophrenia. When a person has COMT VAL variant, then what is happening because of that? In one specific area of the brain, which is prefrontal cortex. Prefrontal cortex means which is right behind the forehead. Their dopamine activity becomes less, and because of that, he has negative symptoms of schizophrenia, not the positive ones. Positive ones come because of there's an area called limbic system emotional, which is below our lobes and all. There where dopamine is more, there we have positive symptoms. Here they're talking about the negative, and negative, like we know, are not the first ones which the psychologist detects. First, he looks out for positive symptoms. So still, but even negative symptoms are important. So what they found in that prefrontal cortex as such, it is that area of the brain which helps us to think, plan, do good mental activity. There their dopamine levels are less. So like we've seen, they become dull, they're not very motivated, they can't think too much because there the activity has become less of neurons. So only in prefrontal. Because the negative symptoms, if we see of schizophrenic, they're related to thinking. Emotional, we have seen as such, they're having too much emotion, sensation and all. But thinking, planning, being able to think for the future, all that is down in them. So prefrontal cortex is that area which helps us to think. So their dopamine is low. And where those hallucination, delusion, and like sensory activities too much, limbic system, there the dopamine is more. So different parts, different levels of dopamine, like that. Okay, so they're telling us further when they have done research, what they found is not only the VAL variant is playing a bigger role. If someone has the VAL variant plus he's a man, so gender is adding to it, then he has the greatest risk of having negative symptoms. If a woman has VAL, even she has risk of negative symptoms, but not as much as a man. So in men, it's playing a bigger role. So all this we have to memorize. Next is about DISC1. That also they have done research and they have found support. So that's also strength of the explanation. For DISC1, what they have done, they took a large Scottish family in which it was known that there are high rates of schizophrenia. So like they've taken a full extended family and they found in that 34 members out of many who are having schizophrenia. They've not mentioned how many, but maybe say there were in 60s or 100 people having schizophrenia. Out of them, 34 were having DISC, one particular gene variant. I think they have not mentioned which is the variant, so we just have to know some particular variant was there. Okay, so that is one. Means if most people who are having schizophrenia are having that variant, so then it's playing a role in schizophrenia, like we always see. They have not gone into the mechanism of this. Then they're telling us more recently they did a meta-analysis of animal experiments. Here we don't need to memorize so much, like broadly we can remember because animal experiments, who they were and all, how many were there, we don't need to know so much. So we can just say animal experiments. Okay, what they found was, in animals, one thing is we can do some types of manipulation. In humans, we just have to study which genes are there, but in animals, we can alter the genes also little. So they're telling they did a meta-analysis of 14 experiments. Meta-analysis means they took many different animal studies that were done, a collection of such studies, and they saw what were the common results. So these are not the results of a single study. They are a result of many studies combined. Okay. So what they found that when they purposely manipulate the DISC1 gene, the presynaptic dopamine regulation becomes less. Okay, here I'll just explain quickly. Other places we are going to see in more details. Again, from biology, you must be remembering that in the brain, we have neurons. So these long type of cells. Now, between any two neurons, always there is a gap, which is called synapse. The cell which is before the synapse, in the left side, it is called presynaptic cell, and which is on the right side, it is called postsynaptic. Now, we don't have to know so much. What we have to know is presynaptic cells, they are the ones which give out the chemicals. So, like the presynaptic cell is creating dopamine and it's releasing the dopamine into the gap. And what the postsynaptic will do, it will receive the dopamine. It will absorb the dopamine which is there in the gap. Here what they're telling us, now if we come back to this, in these animals what they found, this DISC1 gene causes presynaptic dopamine dysregulation. So it's just a technical way of saying that this presynaptic cell which is releasing dopamine, some problem is created in that. So either it is not producing dopamine how much it should, or it is not releasing dopamine into the gap how much it should. It's doing more or less of that. Some problem is there. So they're saying there's a problem with the presynaptic cells. Okay. So this is how they have evaluated. Then we have to look at issues and debates, which we'll do next time. And more I will give in my notes also what we have to evaluate. Okay, do you have any doubts still here? Yes. DISC1 gene, no, it is there in everyone. The variants are different. Yeah. These neurotransmitters are in everyone, dopamine, GABA and all. In most people, it's at normal levels. Those who have disorders, they tend to be more or less in them. Because in some people, they have a variant which is affecting GABA. We are having variants which are not affecting GABA. So the allele is always creating the problem, not the main gene. Which allele or which form of the gene we have, that is determining the disorder. Okay, anything else? Okay, so then we'll continue with this on Friday. Okay, bye then.